Hello, everyone. Welcome to our webinar. My name is Sevana Yaghoubian. I lead the germline applications at SOPHiA Genetics. Today, we'll be discussing how SOPHiA DDM Platform supports the advancement of hereditary cancer analysis through tailored end-to-end genomic workflows. Okay. So today's agenda, I will be walking you through some background and portfolio information and then highlight features of SOPHiA DDM Hereditary Cancer Solution. And my section will be followed by a presentation by Jennie Dring, Principal Clinical Scientist at West Midlands Genomic Lab from Birmingham, where she will be talking to us about the adoption of hereditary cancer V2 into clinical routine in their lab. We will end this session with a Q&A. So key challenges facing hereditary cancer analysis today in laboratories. One is the rapidly evolving landscape. So there is technological changes as well as clinical. There's a lot of guideline changes where there are inclusion of different criteria, as well as inclusion of more genes, as well as complex variants such as CNVs, alloinsertions, and BOLA inversions that are becoming critical to be able to include in hereditary cancer testing. Included in this key challenges are the pressure that laboratories are facing with respect to resource management, turnaround time, as well as adherence to local guidelines. So the key requirements in hereditary cancer analysis today to answer those key challenges are workflows that are consolidated, meaning these are workflows that would actually have one streamlined analysis for SNVs, indels, as well as CNVs and complex regions, which will free up a lot of resource. The other aspect here is the streamlined operations, which means that automation is key now because of the throughput challenges as the number of testing becomes larger and larger in different laboratories. Of course, major requirement is a robust performance where advanced analytical precision as well as sensitivity and good on-target coverage is key for a robust performance of a hereditary cancer solution. In addition to this, which is the key and the last component in delivering cases in hereditary cancer solution, is the simplified analysis. So tools that have simplified analytical workflows for classification as well as visualization in order to be able to streamline the resolving of clinical cases for hereditary cancer. So at SOPHiA Genetics, we have a portfolio that supports and addresses all of these key challenges and requirements for a laboratory that are testing hereditary cancer samples in routine. We have a portfolio where we have applications such as the Hereditary Cancer Solution version 2. These are commercial solutions, and we also have an CIVD product as well. We also have community solutions here. These are applications where we have developed the assay as well as the analytic pipeline in collaboration with KOLs in the field, and these solutions are now being used at multiple locations as well. And we do also support the analytical, with an analytical pipeline, major solution providers for hereditary cancer. And one of the other, let's say, offerings that we have in this area is the custom solutions, where we can customize both the reagent as well as the analytical pipeline to suit the needs of a laboratory. Last but not least, we also have exome solutions where hereditary cancer genes are included, and also they allow for more comprehensive and deep analysis of expanded, let's say, gene lists for hereditary cancer solution. So to date, since the availability of our solutions, we have analyzed more than 170,000 profiles for hereditary cancer solutions through our DDM platform. And we have the solution available in more than 218 institutions globally. Now, a few words about the hereditary cancer solution and what it brings to the laboratories. So one, it's a guideline-driven solution, meaning we have picked 83 biologically actionable genes based on guidelines such as ACMG, NCCN, as well as Panelab. It's got expanded coverage, which means it includes many non-coding promoter regions as well as intragenic hotspots for some essential and key genes. We also have comprehensive variant detection, as I was mentioning earlier. So not only we detect SNVs, indels, but there are CNVs, as well as complex variants such as alloinsertions, Boland inversions, and pseudogene detection of PMS2 and PMS2CL. So in terms of analytics, the interpretation is ready output, which means we have included HGVS compliant variant naming, main transcript, as well as integration with, we have integration with BRCA Exchange and bridges risk scores through the Alamut Visual Plus. What we also used for the development of this solution, and it makes it very efficient, is the fact that it's built on the Universal Library Prep. Universal Library Prep is a reagent set that we have developed where laboratories not only they can consolidate multiple germline library preparations for different applications, but they can also consolidate somatic library prep, specifically when it comes to myeloid solutions, for example, solid tumor solutions, and so on. So this allows the lab to actually consolidate their library prep workflow and again, free up resources. We have also included sample check ID in this application. And here we have a set of SNPs from Pengali et al., which are found in exomes where the user can have an orthogonal testing and then match the SNPs that are detected in hereditary cancer solution V2 to those that are found in the orthogonal test, and then verify the ID of the samples. And of course, the solution, like other solutions from SOPHiA Genetics, is automation ready, which means we already have validated scripts that are available on Hamilton Clinical Starlet, as well as NGS Star, ready to be deployed at any site. Let me just walk you through very briefly through the 83 biologically actionable genes that I was telling you about earlier in the slides. So we have a set of genes that are important in multiple hereditary cancer indications, such as gynecological, prostate, abdominal, endocrine, nervous system, renal, and skin. And you can see the description of the coverage of these genes that are denoted with some symbols up there. So for triangle, that means the gene is fully covered. A square means some hotspots have been included. And the circles, of course, mean CNVs, and that's, of course, available for all of the genes. And then the star represents pseudogene and UTR detection. So now moving to the actual workflow. So as I said, we have a sample to report integrated workflow here. The library prep can take 1.5 days, and then the sequencing another day or two, and then the FASTQ file is ready to be uploaded onto the SOPHiA DDM platform, as well as the subsequent analysis in Alamut Visual Plus. And all this is available with just a few clicks. We also offer integration with LIMS systems as well. So just for visualization, and I want to bring out another point here, that the application is supported by our new web-based platform, the SOPHiA DDM web-based platform, which has a lot of new features that are available now in this web-based platform. And also you have the same simplified workflow where you have the Alamut Visual Plus available through the platform with just a click, just one click. So now I want to talk about a couple of more features about the hereditary cancer solution that are possible through the web-based platform. So the quality indicators, these are very simple indicators where you can see at one glance the quality of your sample. And this is for panel coverage as well as coverage uniformity and on-target rate. We also have CNV flagging and individual CNV reporting and multiple types of files that can be downloaded. Now I will touch upon some of the performance metrics, and later on, Jennie will be talking to you about more details, but just an overall summary. So we looked at about 17 samples. These were clinical samples, and we have the following metrics in terms of performance. The median coverage is around 100%. And the median on-target rate, which is very typical to this application, is around 88%. And we have good performance at 100% and precision for SMVs for 100% at 100% and 98.5% for indel detection. And as you can see, we have highlighted some of the tricky or interesting genes and the variants that were detected. For one of them, most of them, of course, the variant fraction is very strong, and then we have one that is that was detected at a low variant fraction, but this was confirmed by the laboratory that it's a variant that's found at the end of a very long homopolymer, and it's normally confirmed with an orthogonal method anyway. So next, I will just highlight a few, again, performance metrics here with respect to the CNVs. So we looked at many CNVs with our collaborators, and we had good performance. Couple of things to highlight here. We did not detect one in BRCA1 at exon 3. We detected one copy by using reads instead of coverage. And then for PMS2, we had a copy number that we detected, and this is by using our gene conversion module. And you can see that we have where you see the boxes in red, that's what denotes the copy number that is lower than the normal, which is 1. And then lastly, there was one where we did not detect the CNVs, but then by looking at the normalized coverage profile, we could clearly tell that this was a mosaic sample. I would like to conclude with some key messages here from the hereditary cancer portfolio. So, multiple applications that we offer at SOPHiA Genetics that specifically when it comes to hereditary cancer V2, we have a guideline-driven application here where it aligns with most local and international guidelines. It's an efficient solution because, as you saw, that it has a consolidated workflow, sample to report, and it's automation ready, and it's based on the universal library prep that allows for consolidation, both germline and somatic applications. And last but not least, it's supported. And also what is key here for hereditary cancer V2, and as well as some of our other hereditary cancer solutions, and soon our enhanced exome as well, is that you would be able to detect all complex variants through one workflow and be able to visualize them as well through Alamut Visual Plus. With that, I have concluded my section, and I will pass it on to Jennie Dring now to go through her presentation. Thank you.
Great. So thank you for that introduction, Sevana. I'm going to talk through genomic advances in hereditary cancer and optimizing variant detection and interpretation workflows. So here's the disclosure and disclaimer. And so just an overview of what I'm going to talk through today. So I'm going to give a brief introduction, discuss the UK context of our work for those who aren't familiar, the breadth and throughput of our work, our impetus to switch our platform, implementation of SOPHiA DDM hereditary cancer solution, and then I'm going to do a walkthrough of the use of DDM in our hands. I'm going to talk through some of the testing outcomes that we've seen and the impact of implementing SOPHiA DDM. And then I'm just going to highlight some of the migration to the next generation of SOPHiA DDM with some screenshots of what that's going to look like and what's coming next for our laboratory. So just to give an introduction, I'm a principal clinical scientist leading the solid cancer team within the West Midlands Genomic Laboratory. We're a very large team of more than 300 people within the lab, and we serve a population of more than 6 million across the West Midlands. We report about 90,000 tests per year, and we're a clinical laboratory, so we're UCAS and ISO accredited. So as you might be aware, within the UK, healthcare is centrally funded and free at the point of care. So for genomics, that means that we have a National Genomic Service, and this is split into 7 genomic laboratory hubs. Our laboratory sits within the Central and South Genomic Laboratory Hub, and you can see in orange there our geographic coverage. That includes the West Midlands, Oxford and Wessex genomic labs. So the testing that we offered is defined by the National Genomic Test Directory. So this defines both the testing, the technologies to be used, the gene targets, and the patient eligibility for those particular tests. So as I mentioned, we're a really large laboratory, and we cover testing across cancer and rare diseases. Today I'm going to focus on hereditary cancer. So we offer testing for 21 different indications. So that includes some common hereditary cancer predisposition syndromes, such as those associated with breast and ovarian cancer or colorectal cancer, and also some rarer syndromes such as Li-Fraumeni. Our gene panels vary in size from 1 to 13 genes, and we have a wide range of referrers, both from our colleagues in clinical genetics and from mainstreaming through oncologists and the surgical team. So the impetus to switch our platform, so in 2024, CNV analysis for every gene on the test list became mandated as part of the test directory. Up until this point, we've been offering NGS for SNVs, and we're using MLPA for our CNVs. So we want to consolidate our SNV and our CNV workflows together to be able to offer a CNV analysis for every gene on each of our panels. We also needed to be automation ready. And some of our requirements for making the change were that we had a deep, consistent coverage across all 44 genes that are part of our portfolio, that we were able to have efficient variant management and labeling, and that we were able to integrate some variant classification into the system that we were using. So we implemented the SOPHiA DDM hereditary cancer solution in October of last year, and we're using it for all of our inherited cancer referrals. The majority of those are blood samples or DNA received from other laboratories, with a small number of saliva samples as well. At the moment, we're testing between 47 and 94 patient samples per week, and we're running those on a Illumina NextSeq platform at 47 samples in a run. We've tested more than 1,500 patients so far, and we've just transitioned from a manual to an automated robotic setup. In our hands, we've seen a low failure rate, so the samples that aren't meeting our QC after enrichment of 4.15%. So just going to start walking you through what our process looks like. As you might expect in a clinical lab, quality control is going to be what we'd look at first. So SOPHiA DDM has a traffic light system, which you can see at the top, which flags quality parameters that are important for the analysis. So that includes coverage. And then there are also warnings for things like pseudogene regions, or areas of complexity in calling. We use a coverage threshold of 50x, and where we're looking for that up to plus minus 5 bases from the exons. With our using SOPHiA DDM, we've seen a much improved depth consistency, and this has dramatically reduced the number of Sanger sequencing fragments that we're having to do to cover gaps in coverage. So our rescue rate has actually dropped from 22% in some panels to 0.23% across all of the genes that we're using SOPHiA for. We've seen that the platform is able to handle suboptimal sequencing really well, and we're getting some really robust results even when we have that suboptimal sequencing. One of the other key features is that we have transcript level visualization, and that's important to make sure that we're looking at the clinically relevant transcripts. So you can see those drop down in the coverage on the left. So when we look at the data, we get data for every gene that's on the panel, but we are only usually interested in a small subset of that. So we apply a virtual panel. For this patient sample, this is for the breast cancer panel, and this has got 7 genes on it. So you can see on the left, we've got our 7 out of 84 genes, and that's found 113 variants. We're able to apply a custom filter to this data so that we can reduce the number of variants that we want to look at. So our filter defines our region of interest. It also excludes genes that we don't want to analyze report SNVs in, such as GRAM1 or EPCAM, and it also excludes variants that we've previously classified as benign. So in this patient, we've got 5 variants for our analysts to review. So 2 of these with the hatchings are variants that we have previously reviewed and classified as artifactual or false positive cause. If you look at the variant frequency there, you can see that it's not what we would expect for a heterozygous variant, and this is actually within a homopolymer region. So these are quite frequent artifacts that we're able to easily exclude using DDM. So to look at one of those variants in a little bit more detail, you can see we've got excellent depth for this variant of more than 1,000 reads, and that we're seeing it at the 50% expected frequency for a heterozygous variant. We get 3 different indicators for the frequency of the variant: the frequency within the run, within our account, and within the wider community. This is really helpful to identify variants that potentially might be artifactual if they're frequent on a run, or if they're seen more commonly within the account or community, might represent benign or variants or polymorphisms. It's easy functionality for us to see whether we've reported a variant before in the middle, or whether we've called a variant through as an artifact. There are multiple really handy links out that will take you straight to the particular page for a variant. So for Nomad, it links out so we can look at population data, we can look at reports on ClinVar, and then we can also use IGV to visualize that variant if there was any concern about whether it was real or not. We can use the NCBI, Google and OMIM for a bit more information. And we can also open up Alamet Visual Plus directly from the software, so we can review the functional information there, such as the splicing and conservation. However, there is also direct integration of some scores within SOPHiA DDM, so that's including the PhyloP for conservation, and on the web version, the Revel score has also been added. So just to look at what variant classification looks like within the platform. So we're able to see any prior classifications and look at what the underpinning evidence is. One of the key things for our transition was that we were able to have our previous benign variants added before implementation, and that was really key for some highly polymorphic genes like BRCA1 and 2. So you can see in the top view, there's a number of different ACMG criteria that we might want to use for a variant, and then we can apply each of these criteria with reviewing the evidence together, and that enables us to put both the specific criteria together to put the classification. So for this particular variant, it's we've applied BP1 and BP5, and this means that this variant we're classing as a likely benign variant. So looking back to those 5 initial variants we had in our filter, one of these has actually been flagged as benign as part of our analysis, so that's no longer present in the filtered data. So we're down to these 4 variants. We have our 2 artifactual variants, and then we have the likely benign variant that we've looked at, and then a further variant, which is a variant of uncertain significance. So it's really clear with the hexagons on the left to see what our prior classification was and whether we want to report those variants or not. So the CNV analysis is one of the impetus for our change. So in the same patient, we've actually got a variant that has been detected. So if you look at the top view, we can see what's been called. So there's a really helpful box plot visualization of the gene, and you can see highlighted in orange that we've got a additional copy of in 4 different areas. And that's the only CNV that's been called in this patient, and you can really clearly see that in the plot. So you can review that, and also we review that manually for detecting mosaic variants, as Sevana highlighted in her presentation. And so SOPHiA DDM is also able to identify some really challenging variants. So for our portfolio, and that includes the Boland inversion in MSH2, and you can see on the left an IGV screenshot of what that variant looks like. And we've recently also identified a allele insertion within the ATM gene, and that's on the right-hand side there. So we've been able to detect all the variants that we are interested in reviewing. So just to kind of summarize our testing outcomes since we implemented SOPHiA 7 months ago. For our ovarian cancer panel, we've seen an increase in the pickup rate for likely pathogenic and pathogenic variants from our prior workflow at 8.5% to 10.9% with SOPHiA. That's probably due to CNVs in some of the ovarian cancer genes like RAD51C and D that we wouldn't have been detecting using our previous approach. Reassuringly, our VUS reporting rate has remained consistent, so we're not detecting more VUSs with this platform. And we've got a huge increase in the number of genes that we've got CNV coverage from. So with our prior workflow, that was 21, and we're up to 44 genes with this product. So far, we've had 100% orthogonal confirmation for all of our reported findings. So CNV calling is something that's slightly newer to us. We've been seeing a CNV failure rate of about 5.3%. This is where the noise has been too high for successful CNV calling. We are hopeful that with the implementation of automation, we should be able to reduce that. We've seen a much lower noise level once we have in our automated runs, but also because of the different sources of the DNA samples extracted at multiple different labs, this may be something that is just a feature of our patient referral population. And so an overview, we've massively increased the efficiency of our approach. It enabled us to consolidate our workflow. It's reduced the technical hands-on time by moving to kind of one workflow. And we expect that this will be further reduced going forward with a further saving of about 5 hours per run with automation fully implemented. We've seen an improved technical turnaround time. So our turnaround time for urgent cases is moved from a minimum of 8 days to a 4-day technical turnaround time. And what we've also seen is an enhanced variant analysis. So we've detected variants that would have been missed by our previous strategy, and we've been able to integrate variant classification into the platform where it's separate before. So we will be migrating to the new generation of SOPHiA DDM in the future. Some of the advantages of this are that it's web-based, so that will increase our flexibility by helping to support remote analysis. It will also give us flexibility in applying some of the ACMG criteria. We follow the CanVig UK guidance, and we need to modify the weight of some evidence that we apply, and the new version will enable us to do that. It also has further security on the virtual panels with the ability to lock these down further, so that can help reduce the risks of panel modification. And also we'll be able to select upfront the transcripts that are clinically relevant that we are most interested in. So this is a screenshot of the same patient that we looked at earlier for their variants within the web version. So we've got those 4 variants the same. If you look on the right, you can see that our annotations have been pulled through. So we've got those 2 artifactual findings in ATM. And we've got our likely benign finding in BRCA1 and our BRCA2 VUS there. And we can also visualize the CNV plots within the web version, and we can clearly see the duplication that we had previously observed within this patient. So we've got probably even more changes coming soon. We're in the process of deduplicating some of our testing. That will mean that we'll be centralizing the wet lab work in a single location. So we'll be needing to distribute some of our analysis across different laboratories. At the moment, we're exploring the use of SOPHiA DDM dispatch and peer network to facilitate that process in the future. And I'd like to acknowledge the rest of the team. Obviously, I don't do all this work on my own, so our technical team for forwarding all of our wet work, the implementation scientific team, and our bioinformatics team as well.
Thank you, Jennie, for that very insightful presentation, and thanks for being a trusted user of SOPHiA Genetics applications. And now we will open up the floor for questions from the listeners.